Sankofa Arts Trust, in collaboration with Vaccine Resource Group and AVAC, present the HIV Community Research Update as the world observes HIV Vaccine Awareness Day. HIV remains a big challenge, particularly in sub-Saharan Africa. About 1.4 million people live with HIV in Zimbabwe. Out of these, close to 1.2 million are on ART. While ART is currently saving millions of lives, ending AIDS by 2030 can only be a reality if an effective HIV/AIDS vaccine is found, a vaccine that is available, acceptable, accessible, and affordable by all. The sustained control and eventual end of HIV will depend on methods that provide long-lasting protection, including an HIV vaccine. The dramatic increase in global coverage of HIV treatment, care, and existing prevention options must be sustained and expanded to further bring down the new HIV infections and deaths from AIDS. At the same time, research to find additional options to meet people's diverse needs and desires must continue. A safe, effective, long-acting HIV vaccine that is accessible to those who need it the most would make a real impact as part of a comprehensive strategy to end the epidemic. An ideal vaccine would require few shots and last for years, which would be easier to use than many of today's most effective HIV prevention methods that require daily use or regular adherence. In 2009, the Thai vaccine trial known as RV144 showed proof of concept that a preventive HIV vaccine is possible. Researchers have continued to build on the results of that trial to refine and develop potentially effective vaccine strategies. Vaccine research is a long-term endeavor. HIV vaccine research has been underway for more than 35 years, but vaccines for other diseases that are in use today also took decades to develop. This is a time of tremendous progress in vaccine research. Vaccine development is always a long-term process, but we've seen unprecedented advances in recent years, and several promising concepts are being tested in large-scale trials. At present, 2 vaccine candidates and a vaccine-related approach are being studied in large-scale efficacy trials. The HVTN702 trial, known as UHAMBO, ongoing in South Africa, is building on the RV144 trial mentioned before. Results of the HVTN702 are expected in 2021. The HVTN705 or HPX 2008 trial, also known as Imbokoto, underway in multiple countries in Southern Africa, is proof of concept study using a novel vaccine with mosaic immunogens. Led by the pharmaceutical company Janssen in partnership with a number of public and philanthropic global vaccine development partners, the study's results are expected in 2021. HVTN 703 and HPTN 081 in Africa and HVTN 704, HPTN 085 in the Americas and Europe are 2 efficacy study testing antibody-mediated prevention or AMP. In a strategy known as passive immunization, this research is investigating whether direct intravenous infusions of a broadly neutralizing antibody called VRC01 can prevent HIV infection. The AMP trials will also provide insights that can be applied to both passive immunization as a preventive strategy as well as to vaccine development. Study results are expected in 2020. As large-scale efficacy trials move forward, researchers, advocates, and communities are working together to ensure that trials are well conducted, that they conform to good participatory practice guidelines, and also to adapt to changing realities of the HIV response. This includes adding new prevention options, such as oral pre-exposure prophylaxis, into the standard prevention package of ongoing and planned trials. Researchers and stakeholders must also plan together now for acting on the results of these trials. It is important to understand and clearly communicate potential next steps for each of the products and concepts after trials and results to be disseminated. Regardless of results, these efficacy trials will produce data that will move the vaccine field forward. Early-stage research and trials are focusing on improving existing concepts and products as well as developing and testing new ones. BNABs have the ability to neutralize or block block any strains of HIV and potentially prevent the virus from establishing a lasting infection. By blocking many different strains of HIV, these BANABs may help address one of the greatest challenges in HIV vaccine development, which is the virus's ability to mutate rapidly. Understanding how BNABs block HIV when delivered as passive immunization could inform the development of preventive vaccines. Understanding how much antibody is required to achieve protection provides a target for future vaccines. For more information on the state of HIV vaccine research and advocacy, please visit www.avac.org/HVAD. This program was brought to you by the Sankofa Arts Trust in collaboration with the Vaccine Resource Group and AVAC.